Combination between tannin and alcohol. it's again the concentration, because the alcohol that kills the bacteria, 75% can survive it. Like the tannin, you have to drink, for example, a liter of wine to have the effect of the concentrated drug. And yeah, people go to herbs again and tonics, but they don't know what's the effective concentration. And sometimes you can overdose it. And I happen to teach microbiology here at UNM one semester, and I remember this because you have to attract the attention of the students. They're very distracted. So I found online this case of an Asian girl from the Philippines. She had allergies and shrimp as well, but yeah, Asian people like shrimp. So knowing that she has allergies, she ate the shrimp, and then she usually makes a tonic that her grandma used to make, and from some herbs and roots. And then it's a combination of things. It wasn't published what exactly, but after eating shrimp, she got very severe attack, and instead of like taking drug, she made really strong tonic. And then the next day she woke up. That was the nightmare of every woman. Like her face, she was 25, her face looked like 95. And it's all about overdosing. And of course, people started to do analysis of the what's in the tonic, and there were some components that are of the so it makes it's in the creams for allergies. I forgot right now.
Antihistamines?
It's not an antihistamine. It's a cortisone, but it's a plant-based. But they're pretty close in structure. Some estrogens also found in plants, but not exactly the same formula, but very close. So this one, the cortisone analog, when overdosed, it had the same effect. It hit the same receptors, and we know that we are on corticosteroids, our skin gets a little bit more brittle, but it's one dose and like 10 times more. It has this terrible effect of destroying the like elastin, the protein that's between the cell that keeps the cells together. So that was overnight aging. So it kind of tells us that yeah, it's good to use herbal medicine, but in some cases, when you're not quite sure what's the full effect when you overdose it, of course, if it's like plant chamomile, you won't get any negative effect of it. But some other, especially Asian herbal medicines, can have very potent effect, especially when concentrated. So that's why, yeah, contemporary science, there is this link also of drug development is based on herbal derivatives, and then finding the potent element, and then finding the right concentration that's effective for bacteria. But then a new study has to be done for toxicity at this particular concentration on primates and then humans.
How is the lifetime, or how long is the lifetime of a new antibiotic when it comes out compared to the 10-year development phase that you need?
You mean the lifetime when it's potent?
Yes.
So people study in the lab when they discover a new antibiotic, and they start passaging pathogen in the presence of antibiotic. In 1 week to 1 month, they can isolate resistance. So the main idea is that the mutations that cause the resistance to the antibiotic are already in the population. Because bacteria has a huge diversity. They're like 99.9% identical, but the 0.1% mutations, they make them resistant already to something that's new. They haven't never seen it. So the antibiotic starts selecting for it. So when you want to know the statistics on the population of the whole human population, you won't see the resistance probably in, I don't know, I haven't read it, but I would assume within 5 to 10 years, if people are not reporting, because many times you have resistance, but nobody is like studying it and then publishing. But if you do the math based on lab experiments, how many cycles of exposure take to isolate this resistant, probably it's like less than 10 years. But to show in the whole human population, it will take longer. Yes.
I can help you answer that. I serve on the FDA.
Thank you.
A drug, probably a minimum is 4 or 5 years if you have a good drug. You can't take a drug out and give it to people without knowing it doesn't kill people. So you go through actually 3 phases of studies. You first go through phase 1, which is what Sofiya is doing, finding out if it's a drug, what it does and what it works and what it doesn't do. And then they do a phase 2 study where they pick small groups of people, maybe 50, maybe 100, who have the disease and try the drug on them, try to dose it and see how much dosing they're having. When they think they have a drug and they have a good dose and they don't think they have side effects, then they go into a phase 3 study. Most phase 3 studies have at least 1,000 patients because people are going to be different. We aren't all the same. Some of us are going to get a kidney disease from a drug and some of us are not. And so they're going to try that for patients that will watch them for at least a year on this drug before they release the drug. And if you get 2 good phase 3 studies, then you can go to the FDA and ask for an approval. And the FDA, if it looks like a good drug and has no side effects, they approve it. If it's a drug that may have some side effects, maybe not, you have to decide, then you call a hearing. And the hearing committee will give advice to the FDA whether that drug should be released or not. And often it means go back and do more studies. Maybe you ought to check it more on people that have certain diseases or certain types before you release the drug. So with that, I think the short answer is anywhere from 4 to 10 years.
But it could be a race if something is developing resistance in 5 years and it takes 6 to deliver, we might start losing that race.
That's right. We are losing that race now. The new antibiotics that we're developing are antibiotics that are very specific to just a few enzymes. We've gotten all the easy ones already. I mean, our penicillin, our tetracyclines, our sulfas, we're wearing those out. And I think it is misuse of antibiotics that's causing some of our antibiotic development. And that's how you can see it. You take chickens and give them an antibiotic, and pretty soon they've got bugs that aren't killed by that antibiotic. So part of it is we're going to need to find things other than antibiotics that kill bugs.
Yeah, or conventional antibiotics, because once they start killing bugs, we call them antibiotics. Tannins are not antibiotics yet.
So, but then if that's the case, should maybe we be focusing research on enhancing the immune system, doing more to enhance like interleukins or, different, whether it's memory cells or whatever, to just try to get the host to kill stuff?
Yes. Actually, DITRA also has calls for those alternative treatments. The immune system, stimulations of the immune system. And for some pathogens it works. For others, stimulating the immune system makes them even more aggressive. We tested vitamin B3, nicotinamide, to chase out the persisters, this dormant population, because it boosts the energy of the cell, not just our cell, but bacterial as well. And then we found studies that have been done with Helicobacter puri, that's the one that is in our stomach and causes ulcer, and also with Staphylococcus aureus that usually causes infections of the upper respiratory system. And it helps. It helped. First, one study was with no antibiotic supplements. It helped and it was done in mice, of course, not higher animals, but it helped the animals to survive when, like 50% of the, it's a good hit. With antibiotics, it even had better effect. we tried one of the exotic bacteria, like this Barcodera pseudomaliae, because Dieter was interested in this one. It has been developed as a bioweapon early in the races between the two systems, and it has never been released, but still there is a threat, and they want to find countermeasures. And also it's bad bug for East Asia and then Australia, but also our troops are deployed everywhere. And we tried it, and it had the opposite effect. It actually killed the animals faster than just treating them with salt, and then salt plus antibiotics. But even in combination with antibiotics, it became, the animals survived, but they were sick, and then the bacteria came back. And because each bacteria has different adaptation, like weaponry when they attack our system. Some of them, or probably our immune system reacts differently. And it all depends what's on the surface of the bacteria, what fatty acids make the system assault the innate immune system. And this particular bacteria is causing very strong activation of the first responders, the white blood cells, the neutrophils, and usually the neutrophils are also killing the host cells, and they're opening kind of channels for the bacteria to get in. And also all this inflammatory response that starts from the neutrophil activation. And it turned out also that the vitamin B3 activated the neutrophils, overactivated them. So they started to really, so there is always a balance, and the problem is there is no silver bullet for each bacteria or pathogen. It may work for some, not for others, and but people are, there is like money coming into science from Department of Defense to find alternative methods just to help the immune system.
So what I've read is that around 1917, when the influenza outbreak, that the world's population actually went down because of the epidemic, not because of the war. Do you see a huge incident like that in the next 50 years?
I don't know. It all
where we are
depends.
headed?
Yes, so it's virus, and you have seen movies that are apocalyptic movies, and the ones that was very strong also in my mind was the Contagion. The name of the movie, I don't know if you have Contagion or what's the Contagion. Contagion, okay. That was also about influenza virus, and so what happened first? It was a time when people were very few people could eat well, and their immune system wasn't very strong. So now we are in a better situation. Our immune system is much stronger. Also, it was very new, so the immune system did not have any defense against it. Now, this particular virus is part of the basic of the vaccines. But people predict, they do those kind of mathematical calculations to see what can happen. So 1 in 100 years, so we're overdue of this type of infections. But the problem is, not the problem, the good thing is, as I said, we have a stronger immune system now. Also, most of the knowledge of how to deal with overreaction of the immune system is also there in the medical community. So most of the mortal cases are not caused by the viral infection, but by the immune system overreacting to the virus. And if you see even nowadays influenza, people that are the most vulnerable are either kids or young men, 25 years old, if they're told the worst. And then people after their 60s or 70s or 80s. It's all personal, depends like personal health. But and then there are two different reasons. For the young and elderly, it's the immune system that For the young, is not well trained. For the elderly, it gives up because our thymus is shrinking and losing power. For the young men, especially tall ones, it's the deal with so their temperature rises very fast, and it causes all the damage. The temperature control, scientists have found that in this particular young group of people, so it's when doctors know how to deal with those like responses of the immune system, like draining the body, not draining it, but giving more fluids, and also like bringing the temperature really down. So all those advances, they weren't there during the 1918. So people were on their own with their immune system.
Could you pass that map around?
Yes, so that was a map of the multi-drug resistance. And usually it's, where it hasn't been found, it's deserts or countries where it's a failed state, so nobody reports. There is no administration to take care of the like reporting the cases to the global health organization. But interestingly, the entire Asia is like in the dark hours where it's like more than 5. So a bug found to be resistant to more than 3 antibiotics at the same time. And and those are individual cases. It's not like everybody got it.
Right.
It's just based on cases. And this is the good thing is that usually those multi-drug resistant bacteria, they are not passed like the plague used to be. So they're contained to a particular individual. And why the Department of Threat Reduction is sponsoring science like this is to be able to respond to biothreat or bioterrorism when specific organisms have been developed to pass really fast through like touch and saliva, and then they also carry lots of drug-resistant genes. But usually bacteria is not so dangerous as viruses, and very few is known about the viruses. Only those that are like persistent in our populations, that those are the most studied, and antibiotics have been developed against them. And I'm also part of a project that has been sponsored by the lab research and development money that looks for, and that young gentleman is also Omar, part of this. So just looking for the interaction between the influenza virus and the human cells, how it makes the cell less responsive to infection, and if we find proteins that are very consistent between those interactions, then drug development starts from there, like screening for new drugs and approving them.
So did I understand you right that you're going after it in a different way of cutting the RNA instead of binding to receptors? Is that kind of what you're saying?
No, just going after an enzyme that has multiple functions that are vital for the bacteria. Not like nowadays antibiotics are targeting a single target. And then when, even if the antibiotic loses a little bit the grip on this target, it's still functional and the cell can continue. It can be slower but continues to grow. We're looking at target that's vital because it has multiple functions, and one is to make the right size of the ribosomal RNA, but it also keeps the protein levels of particular toxins. So some proteins in bacterial cells, even in our cells, in higher levels they become toxin. In lower level they have some function. And usually those are very fast cleared out of the cell by enzymes like this one. So if the enzyme function gets slow, not completely inhibited, the accumulation of those toxic proteins will kill the cell. So we call it suicidal.
And so in this test where you're testing all these different things, how are you discovering that they're binding to these multiple places?
Yes, we design a test where we add the enzyme and then we add the target of the enzyme. And now we figure out that the target has to be much more complex in structure to what people have done before. Because this enzyme, people have shown interest in looking for inhibitors since 70s. No, 79. But still no inhibitors have been found. And it turned out that they have been using very simple